Hello, and welcome to Science with Mr. Morgan. In today's episode, we will be learning about mitosis and meiosis. So, to start off, let's look at mitosis from a broad perspective. Remember, the DNA is organized into chromosomes, but it only winds into these chromosomes when the cell is ready to divide during mitosis. When mitosis begins, DNA in the nucleus wraps around proteins to form chromosomes. Each organism has a unique number of chromosomes. Humans have 23 pairs, which is 46 total. Now, when DNA is replicated, it forms a chromosome made from two identical sister chromatids, which makes an X-shaped molecule. These two chromatids are held together on the chromosome by the centromere. This is the same location where spindle fiber microtubules attach during mitosis, which separate the sister chromatids from one another. It's kind of like X marks the spot right in the center of the X. During mitosis, the two chromatids must be separated into separate chromosomes, which makes each chromosome half of the X. Each daughter cell produced during mitosis receives one copy of each chromosome, aka each half of the X goes to a different cell. Now that we have a general idea of what mitosis is, let's look at it a little bit deeper. So mitosis itself is split into 4 phases. The first phase is prophase. During this phase, chromatin, which is just unwound DNA, condenses to form chromosomes. It winds up super tightly, so tightly that you can see it under a microscope, but when it's unwound, you can't. The nuclear envelope, which is the membrane surrounding the nucleus, disappears in this phase. Spindles then form and attach to the chromosomes to help them move to where they need to get to. The second phase is metaphase. And during this phase, chromosomes line up in the center, or the equator of the cell, in a row, one on top of the next. They're moved with the spindles. The third phase is anaphase. During this phase, the two sister chromatids of each chromosome separate as the spindles pull the chromatids apart, like I referenced earlier. The end result of this is that there are 2 sets of identical chromosomes. The fourth phase is telophase. Now, during this phase, the spindle dissolves and the nuclear envelopes form around the chromosomes in both cells. After telophase, each new nucleus contains the exact same number and type of chromosomes as the original cell, aka the parent cell, and it is ready for cytokinesis, which means cell movement. During cytokinesis, the cytoplasm divides and the parent cell splits into 2 genetically identical daughter cells that each have their own nucleus. After cytokinesis, a new cell membrane forms. And if this process is happening within a plant cell, a new cell wall also forms, but animal cells don't have cell walls. One parent cell results in 2 genetically identical daughter cells via mitosis. Now, mitosis is similar to another process known as meiosis. However, it's important to note that there are some significant differences between these two. So before we get into the specifics of meiosis, let's look at it from a broader perspective, just like we did with mitosis. During sexual reproduction, which we talk about in another episode, gametes from 2 parents are combined. What are gametes, you might be asking? They are reproductive cells, such as sperm cells and egg cells. Gametes must have half of the number of chromosomes as a typical cell, otherwise there would be too many chromosomes present when they combine. The zygote, aka a fertilized egg, must contain genetic information from both parents, so the gametes have half of the typical number of chromosomes, which ensures the offspring gets half of its chromosomes from each parent. Half of a set of the required number of chromosomes plus another half of a set of the required number of chromosomes equals 1 full set of chromosomes. Like I mentioned earlier, different organisms have a unique number of chromosomes, and human cells have 23 pairs of chromosomes, or 46 total. Now, each one of these is a homologous chromosome. This means that these chromosomes are the same length and have the same centromere position. For each pair, one chromosome comes from mom, and the other comes from dad. These chromosomes have the same genes, but they can have different alleles. So what are alleles? Well, alleles are alternate forms of genes found on chromosomes. For example, eye color stems from different alleles. That's just one example of many. At this point, we've talked about how gametes have half the number of chromosomes as a regular cell. There are special names for this, and they are haploid and diploid. Diploid cells are cells with two sets of chromosomes. This is represented using 2n, where n is the number of chromosomes. Most human cells are diploid, with the exception of gametes. Haploid cells are cells with one set of chromosome. This is represented by n, where n is the number of chromosomes. Sex cells, aka gametes, are haploid. They're the only haploid cells in humans. When two haploid cells combine, a diploid cell is formed. A haploid sperm, which is 1n, plus a haploid egg, which is also 1n, come together to form a diploid zygote, 2n. So now that we have a general idea of what meiosis is, let's look at it a little bit deeper. So before meiosis begins, DNA replication occurs, so each chromosome contains two sister chromatids that are identical to the original. Meiosis itself happens in two separate divisions, which are identified as meiosis I and meiosis II. Each of these has the same phases. Just like mitosis, prophase, metaphase, anaphase, and telophase. Again, similar to mitosis. Cytokinesis occurs after telophase each time, so also similar to mitosis. Here's what's different though. Between meiosis I and meiosis II, DNA replication does not occur. This is a large difference from mitosis. In meiosis, one diploid cell, aka the parent cell, results in 4 haploid cells, aka the daughter cells. So the first part is meiosis I. In this part, pairs of homologous chromosomes are separated from each other, and they must line up in their homologous pairs during metaphase I. The first phase of meiosis I is prophase I. During this phase, homologous chromosomes line up together and cross over, which is the exchange of DNA between homologous chromosomes. This forms new combinations of alleles on the resulting chromosome. Spindles form, chromosomes condense as they coil up tightly, and the nuclear envelope disappears, all within prophase I. The next phase is metaphase I. During this phase, homologous chromosomes line up in their pairs in the middle of the cell, like I mentioned a few moments ago. Chromosomes that were passed from the mother or the father can each attach to either side of the spindle. Now, it is important to note that we are not talking about sexual reproduction or fertilization here. Instead, this is just referring to chromosomes that were passed on from the parents of the individual that is undergoing meiosis. Let's use Johnny as an example. Johnny's body is undergoing meiosis to make gametes. His mom and dad passed on their chromosomes onto him, and now those chromosomes are being mixed up during metaphase I. So a little bit of his mom and a little bit of his dad. Now back to spindle attachment. This attachment is random, so all of the chromosomes from the mother or father do not end up in the same gamete. This means that the gamete contains some chromosomes from each parent. Some of the chromosomes that our example Johnny got from his mom will be mixed in with some of the chromosomes that Johnny got from his dad. With the number of chromosomes that humans have, there are over 8 million different combinations of chromosomes that are possible within a human gamete. So after metaphase I comes anaphase I, and remember we're still in meiosis I. During this phase, homologous chromosomes are separated as the spindles shorten and they begin to move to opposite sides, aka opposite poles, of the cell. After anaphase I is telophase I. During this phase, the spindle fibers dissolve, but a new nuclear envelope does not form. This is also different from mitosis. After cytokinesis, the nucleus will immediately begin to divide again, so there is no need to form a new nuclear envelope. After telophase I, cytokinesis occurs and the original parent cell divides into 2. And when I say a parent cell, I am not referring to Johnny's parents this time. Instead, I just mean Johnny's original cell that was undergoing meiosis as the parent cell. Mitosis only goes through one cycle, while meiosis undergoes a second. And this is known as meiosis 2. During meiosis 2, sister chromatids are separated and the gametes are produced. So meiosis 2 starts off with prophase 2, just like meiosis 1 started with prophase 1. During prophase 2, the chromosomes condense. Followed up with metaphase 2, the chromosomes line up one on top of each other along the middle of the cell, just like they did in mitosis and just like they did in metaphase 1. The spindle is attached to the centromere of each chromosome. Once metaphase 2 is done, the cell enters anaphase 2. During this phase, the sister chromatids separate as the spindle shortens and move to opposite ends of the cell, just like they did with anaphase 1. Next is telophase 2. During this phase, a nuclear envelope forms around the chromosomes in all 4 cells. After telophase 2, the cell undergoes cytokinesis. During this, the 2 daughter cells produced in meiosis 1 split into 2 more daughter cells, resulting in 4 haploid daughter cells. So you started with 1, it results in 4. Now that we have covered mitosis and meiosis, let's summarize the differences between these two. So mitosis is used to produce new cells, while meiosis is used to produce gametes. Mitosis results in 2 cells, meiosis results in 4 cells. Mitosis undergoes 1 round of cell division, but meiosis undergoes 2. Mitosis results in diploid cells, while meiosis results in haploid cells. In mitosis, the daughter cells are identical to the parent cells, but in meiosis, the daughter cells are not identical to the parent cells. They have half of the genetic information. In mitosis, the daughter cells are identical to each other, but in meiosis, the daughter cells are not identical to each other. And that brings us to the end of today's lesson. Until next time.